Good morning, students. So we are going to start now with the exact mechanism of replication, means DNA replication. So here I have made it into two parts where first let us discuss what all the requirements which are required for the natural DNA replication to take place. We shall discuss here, and later on we will focus on to the mechanism of DNA replication directly. Okay, so that is why I have divided this into two parts. So first let us discuss now what are the requirements. As you know that it is a very complex process. The DNA replication is a very complex process and a very quick process, but it is very complex process. Okay, as DNA is held in a chromosomes, so that it takes lot of you can say machineries and enzymes get involved into it. So let us learn what all that. Okay, first one by one. So let me tell about these the requirements. First, the DNA replication to begin, the DNA requires origin of replication. It is also known as shortly as ORI site. ORI site stands for origin of replication initiation site. Okay, so the DNA which is undergoing the replication must and should have with this origin of replication initiation site or origin of replication site. Okay, until and unless this site is found or recognized by the enzymes like helicases or unwindases, the replication never going to begin here. So what is then origin of replication? It is a specific point at which the DNA replication is going to begin. So it is called ORI site or origin of replication. Now the mode of replication. So mode of replication is going to decided based on the number of origin of replication or sites or ORI sites. Okay, so as we have here the prokaryotes and eukaryotes, as the prokaryotes have got the circular DNA, as the prokaryotes have got circular DNA, and there will be only one origin of replication. So as the prokaryotic DNA is circular, there will be only one, at one point there will be only one origin of replication. As they have as they have only one point of origin, so their mode of replication will be unidirectional. That means that replication either has to begin in clockwise direction or has to begin in anti-clockwise direction, in either in this direction or in other direction. So this mode of replication is called unidirectional replication. So remember here, for the point of your competitive examination, it is very important you need to remember in prokaryotes, as they have got the circular double-stranded DNA, will have only one point of origin of replication, and hence they show unidirectional mode of replication. So similarly, in case of eukaryotes, as they have double-stranded linear DNA and they have got multiple number of origin of replication initiation sites. So hence in two direction, means either in the upward direction or in the downward direction, the replication may takes place in the eukaryotes. So hence in case of eukaryotes, the mode of replication will be bidirectional. Why it is bidirectional? Because they have got multiple number of origin of replication sites. Okay, now we shall move on to the next requirement. That is, we shall move on to the rate of polymerization. What is this rate of polymerization? So rate of polymerization is nothing but it is the speed of polymerization. At what rate, at what is the speed of the replication? So replication is a very quick process wherein which it is going to complete the replication of a one chromosomal DNA is going to complete within a few seconds. So for example, here I have taken some of the examples. So replication is a very quick process, as I said, and in case of E. coli, as it has 4.6 into 10 to the power of 6 base pairs, the replication of this amount of DNA is going to take place just within 38 minutes. Okay, so similarly, if you take a human, the diploid, means a somatic cell DNA, where it has 6.6 into 10 to the power of 9 base pairs, the replication process of the entire DNA is going to complete within 9 hours and 15 minutes. So this indicates that the rate of replication is approximately 2000 base pairs per second. Remember this point, it will be asked in the examination once again in CET or NEET examination. So what is the rate of replication? So rate of replication is nearly or approximately 2000 base pairs per second. Okay, now where is the replication takes place? This is also very important for the point of competitive examination, as I have already explained with respect to cell cycle. Site of replication, if you take the prokaryotic cell, as in the prokaryotic cell where they do not have a well defined nucleus, the genetic material is freely scattered in the cytoplasm. The site of replication, in case of prokaryotes it will be cytoplasm, also known as cytosol, whereas in case of eukaryotic cell the replication occurs in the nucleus, because there is a well defined nucleus, and inside the nucleus this DNA is held in the chromosomes. So in the chromosomes, as chromosomes are situated in the nucleus, the replication occurs in the nucleus in case of eukaryotes. So, and as well as when it occurs, if you take, it occurs in the S phase, during the S phase of interphase of cell cycle. Okay, so replication, as I mentioned, it is very important and very essential for the life of a cell or an animal or a plant. Why? Because if the replication takes place and the cell division does not occur, then it leads to a complication such as polyploidy. So that is increase in the chromosome number becomes, so that condition, increase in the chromosome number, so like haploid, diploid, triploid, what you call, so increase in the haploid number of chromosomes is called polyploidy condition. Now another important requirement for the DNA replication is activated substrates. So what are these activated substrates? So substrates are nothing but here deoxyribonucleotide triphosphates. As we all know that DNA is made up of 2 polynucleotide strands, and DNA replication is nothing but the DNA polymerization here. So as it is the formation of the polymer of a deoxyribonucleotides, so deoxyribonucleotides serve here as the monomers. Okay, and that itself are nothing but the substrate. But these deoxyribonucleotides, as there are 4 types, like adenosine monophosphate, guanosine monophosphate, thymidine monophosphate and cytidine monophosphate, and all these 4 types of monophosphate forms of nucleotides which are present in the nucleoplasm. Hence the nucleoplasm is considered as a nucleotide pool with respect to replication here, and they are in inactive form. So replication is an expensive process. It needs much more energy here. It consumes the much more energy here. So hence the deoxyribonucleotides are made to be activated here. So made to be activated to serve as the activated substrates. So how they are getting activated? So just an example I have given here, AMP, adenosine monophosphate, it undergoes phosphorylation with the help of ATP, the energy currency of the cell, in the presence of an enzyme called phosphorylase, to form deoxyriboadenosine triphosphate and remaining one inorganic phosphate. Okay, so like this, all the 4 different types of monophosphate form of nucleotides, deoxyribonucleotides, undergo activation process and get converted into deoxyribonucleotide triphosphates, and these serve as activated substrate here. So you have to listen this carefully and you have to watch it carefully, so that only you can get the ideas that how the activated substrates are made available during the process of DNA replication. Now we shall discuss about the important few enzymes which are involved in the DNA replication. So this point is also very important in your examination. For 2 marks there will be a question. Okay, so there are few enzymes which are required or which are involved in the mechanism of DNA replication, as they are very important in the replication process, and they are also very important for the point of your examination. A 2-mark question will be asked in the examination. List the different types of enzymes involved in the DNA replication. You need to list them in this way. So there are certain group of enzymes, actually not the 1 or 2 enzymes. These are the actually group of enzymes. So once again we are going to discuss in detail with an example for each. So let me first list out all these enzymes, as there are 4 types. You need to list in the same way. Okay, then only you will get the full marks in the examination, because each is going to carry the half marks in the exam. Okay, then what are that? Helicases or unwindases. The first group of enzymes are helicases or unwindases. Along with that, one more group of enzymes called topoisomerases, or also known as gyrases. Okay, topoisomerases or gyrases. And another group of enzymes are DNA dependent DNA polymerases. So as DNA replication is a DNA polymerization process, so they require DNA dependent DNA polymerases, the group of enzymes. Then also the DNA replication also requires an enzyme called primase. So it is also commonly known as RNA polymerase. So primase is nothing but an RNA polymerase. So as we have here DNA polymerase which polymerizes the DNA, here we have RNA polymerase, so which polymerizes the RNA strand. Okay, so primase is also known as RNA polymerase. Okay, then we also have the one more group of enzyme which is very much essential in the DNA replication, as well as very much useful in the recombinant DNA technology, which is nothing but DNA ligase. Okay, so what are the DNA ligase? They are the group of enzymes which join the 2 DNA fragments, as in case of recombinant DNA technology, where the desired gene is going to be joined with the cloning vector DNA in order to carry from the organism of interest to the organism of source, or organism of source into the organism of interest, so that they are used very commonly in the field of recombinant DNA technology. Now let us move on to the discussion of about the role of each of these enzymes. So first category of enzymes, what I mentioned here, it is helicases, also known as unwindases. So what are these helicases? As the name indicates itself, that they are helix destabilizing enzymes. So that means they are uncoiling enzymes. As you know that the DNA is always in the form of double helical structure. These enzymes, when they get bind to the DNA double helix structure, they unwind the 2 strands of the DNA, means they separate the 2 strands of the DNA. So hence they are known as helicases or unwindases. Unwinding is nothing but separation of the 2 strands of the DNA. So separated strands are stabilized after once the 2 strands, here you can see here in the diagram, here it is the helicase or the unwindases, the enzyme. As soon as it recognizes and bind to the DNA fragment, so that part of the DNA, the 2 strands get open. Okay, so each one of the separated strand immediately made to be stabilized with the help of single-stranded binding protein. So what happens if it is not made stabilized? Because as these 2 separated strands are complementary to each other, they once again recoil and form a double helix structure. So hence the DNA replication will be prevented. So as a result, to prevent this, what is being used here? Single-stranded binding protein, which stabilize the single-stranded separated DNA strands, okay, to allow the DNA replication. Then here each one of the separated strand act as a template. So each one of the separated single stranded DNA act as a template for the synthesis of new or the complementary strand. So this is one template, this is another template. On each of this, the new complementary strands will get synthesized. Hence from each one DNA molecule, 2 daughter DNA molecules will be synthesized. Now we shall see the another group of enzymes which I have mentioned along with this helicases or topoisomerases. Okay, so topoisomerases or gyrases. So topoisomerases are the enzymes which cause the nick or the breakdown and reseal the one strand of the DNA and to release. Why they do like that? See, as we pull the two strands of the rope, okay, what happens? At the lower end, the rope gets coiled more and more due to the stress which is applied from the upper side. Okay, so as a result, so that tension or the stress which is creating at the other end, okay, to release that, these topoisomerases are the enzymes which release. What they do, they cut one strand of the DNA, okay, and that will be rejoined once again. So such enzymes are called topoisomerases or gyrases. So topoisomerases are such enzymes in case of eukaryotes. The gyrases are the such enzymes in case of prokaryotes. You have to remember for the point of your examination, competitive examination I am telling here. So topoisomerases are the enzymes which are helix or you can say super helix stress relieving enzymes found in eukaryotes, whereas gyrases are the enzymes which helpful in the super helix stress, okay, in the release of super helix stress in case of prokaryotes. So please remember this. And once the two strands get separated after the relief of this tension or stress, okay, once again those single stranded DNA strands also should get binded with the helix destabilizing protein. So what happens if this protein does not get bind? Once again, as the two strands are complementary, they once again recoil to form a double helical structures. To avoid that, here the helix destabilizing proteins get bind to the DNA strand where it is get into separated. Okay. Then, so we have these important enzyme, that is the next third type of enzyme what I mentioned is DNA dependent DNA polymerases. DNA dependent DNA polymerases. So there are three types. One is DNA polymerase 3, another one is DNA polymerase 2, and the third one is DNA polymerase 1. So these are the DNA dependent or the DNA polymerases which are found in prokaryotes. So same enzymes are going to vary in case of eukaryotes. So same enzymes are referred in case of eukaryotes as alpha, beta and delta enzymes. Okay, in case of eukaryotes, same enzymes DNA polymerase 3, 2 and 1 are referred in case of eukaryotes as alpha, beta and delta enzymes. Now let us see what is the role of each one of the enzyme. DNA polymerase 3, as I mentioned, it is a DNA dependent DNA polymerase. Okay, it is the major DNA polymerizer. You have to remember for the point of your competitive examination as well as for your board examination. For 1 mark question may be asked, name the DNA dependent polymerizer or the major DNA polymerizer is the DNA polymerase 3 enzyme. Okay, this is the one enzyme which is going to complete or which is going to completely involved in the entire DNA replication, you can say. So hence it is considered as major DNA polymerizer. Then the next enzyme is DNA polymerase 2. So this here I have mentioned it as a proofreading or repair enzyme. So after the end of every round of the replication, there may be the chance for the misplacement of the DNA or deoxyribonucleotides. So those mispairing base pairs are identified and replaced with the correct matching type of nucleotide base pair. So that is going to be done by DNA polymerase enzyme, is going to proofread or cross check for the miss base pairs. Okay, so wrong base pairs are going to be identified and are going to be repaired and replaced. Okay, so hence it is known as repair enzyme. Okay, and one more enzyme, that is DNA polymerase 1. During DNA replication, while initiation of the replication, some of the RNA strands, a short RNA strand is going to be added in the initiation of replication. replication. So at the end of the DNA replication, the RNA should not be found in the DNA structure. So hence that RNA primers they are called, and those RNA primers are removed and replaced by deoxyribonucleotide triphosphates, and that purpose this enzyme is going to serve. So both of these enzymes acts as repair enzyme. So if you think this is the major repair enzymes, what is that? That is DNA polymerase 1. It has to do its activity compulsorily during the DNA replication. So hence it is one of the major repair enzyme we can say. Whereas DNA polymerase 2, it is going to proofread, or it is going to cross check for the wrong base pairs that may be there, may not be there. So hence we can consider this as a minor repair enzyme. Sometimes in the competitive examination they'll give you, so which among the following is a major repair enzyme and minor repair enzyme. So minor repair enzyme is the DNA polymerase 2. The major repair enzyme is DNA polymerase 1. As I said, so there will be the addition of RNA nucleotides during the DNA replication that has to be removed and replaced. Okay, so hence its activity is compulsorily needed here. So hence it is considered as a major repair enzyme. Now we shall move on to the another requirement. So as I mentioned, the third category of enzyme is primase. So primase is also known as RNA polymerase. So this RNA polymerase is an enzyme which synthesizes a short segment of RNA, okay, is going to synthesize a short segment of RNA called primer, okay. So that primer is going to be synthesized depending on the template DNA, the template stand of the DNA. Hence it is considered here as DNA dependent RNA polymerase. So is going to replicate it on the new strand at the 5' end depending on the template DNA strand. Hence it is DNA dependent RNA polymerase enzyme. So what is a primer here? Primer meaning is beginner. So it is the primer is a short segment of RNA which is required to begin the DNA replication. So it is synthesized by an enzyme called RNA polymerase or the primase, okay, at 5' end of the template DNA strand, okay, to begin the DNA replication. Okay, then this what happens after the addition of the primer. So primer, as I said, short segment of RNA, so is going to provide the 3' hydroxyl group to initiate the DNA polymerization by the enzyme DNA polymerase 3 in 5' to 3' direction to begin the DNA replication. Okay, so what is the role of primer here? In order to provide 3' hydroxyl group for the initiation of DNA replication by the enzyme DNA polymerase. Okay, then next one, DNA ligase, as the last enzyme. As I mentioned, DNA ligase, they are the enzymes which are used to join the two DNA fragments. How they join? By sealing the sugar phosphate backbone. So what is found in the sugar phosphate backbone? By sealing their phosphodiester bonds. They are going to establish a phosphodiester bonds between the fragments of the DNA. So thus the small fragments are joined together to form a continuous stretch of DNA, and such enzymes are called DNA ligases. So as the meaning of ligase is to ligate or to seal the ends of the two fragments of the DNA. So that DNA ligases are the enzymes which joins the two DNA fragments by sealing their sugar phosphate backbone. Okay, and finally we'll have here the template. So what is the template here? Template is nothing but each of the separated single-stranded parental DNA. So after the helicases are going to bind to the DNA at the origin of replication site, as the two strands start separating, unwinding, each one of the separated DNA strand act as the parental DNA, act as the template. So which is available as a single-stranded, as in the single-stranded form for the synthesis of complementary complementary strand or the new strand. So complementary or new strand, we can say the newly forming or newly synthesizing strand, as it will be complementary to the template strand. We can also call it as the complementary strand. Yeah. So this is how the replication is going to begin. Now we shall move on to the mechanism, exact mechanism of replication of DNA. And before that, let me tell you once again. So if imagine this is the parental DNA, which has both the strands in red color. Okay. So as soon as the replication is going to begin here, the helicases or the unwindases are going to recognize the origin of replication point here, and they bind to the origin of replication point. As soon as they bind to the origin of replication point, they start moving along the double helix structure of the DNA. As they move along the double helix structure of the DNA, the two strands get separated. The two strands of the parental DNA get separated by breaking their hydrogen bonds present between the nitrogen bases of the opposite strand, and they are going to available in the form of single stranded template strand. Each separated parental DNA strand acts as here template strand, on which the new or the complementary, which is in blue color, is going to be synthesized. Okay, so here the same diagram little bit I will modify, and I will give you the with the help of the self drawn the diagram. So you need to practice this diagram. It will be asked in the examination once again for 3 marks to draw a neat label diagram of replication fork. So here, as soon as the unwindases or the helicases enzymes bind to the origin of replication region, the two strands of the DNA start unwinding. As they start unwinding, they are going to result the formation of the Y-shaped structure. So that Y-shaped structure is nothing but the replication fork. Okay, now we shall see, students, the exact mechanism of DNA replication. Okay, so as I mentioned already, that replication in case of eukaryotic cell always begins during the S phase of interphase of cell cycle. Okay, then also I mentioned that replication always begins at a specific point on the DNA molecule. So this is the parental DNA molecule. At a specific point on the DNA molecule called origin of replication. Okay, so first this origin of replication will be identified and or recognized by the helicases or unwindases, the enzymes. They get bind to it and they start moving along the strands of the two strands of the DNA helix. Okay, so as soon as they start moving along the two strands of the DNA, then the two strands slowly get separated. So the helicases or unwindases are the enzymes which bind to the origin of replication initiation site and start DNA unwinding, okay, or separation. Okay, so next, as soon as the helicases or unwindases bind to the enzyme, the two strands get separated. As the two strands, the separation of the two strands move slowly from one end towards the other end, okay, it leads to the formation of a Y-shaped structure, and that is called the replication fork. Okay, how the replication fork will get formed here? As the separation of the two strands proceeds slowly from one end to the other end, it results into the formation of the Y-shaped structure like this, and that is called replication fork. So you will be asked in the examination to draw a neat label diagram of replication fork. So you have to draw the diagram like this, and you need to have all these labels in a diagram. Okay, then once the replication fork is formed here, each separated strand in a parental DNA act as template. Each separated strands in a parental DNA act as a template, or we can also consider it as old strand. So the template here in this side, DNA, it will be in 3' to 5' direction, whereas on the other side the template is in 5' to 3' direction. Okay, so on each of the template the DNA polymerization occurs. Okay, on each of the template stand the DNA polymerization occurs. Okay, so DNA polymerization always occurs in 5' to 3' direction. You need to remember here, DNA polymerization occurs always in 5' to 3' direction. It never occurs in the opposite direction. So must and you should remember, this is very, very important. The direction of polymerization is very important to remember, that is always occurs in 5' to 3' direction. And this DNA polymerization takes place by the enzyme called the major DNA polymerase, as I mentioned in the last case, that is DNA polymerase 3. Okay, so major DNA polymerization occurs by the DNA polymerase 3 enzyme. Okay, so to begin the DNA replication on each of the template here, what is required? RNA primer is required to begin the replication on each of the template. So what is required? RNA primer is required. So that to begin the replication here, the small stretch of RNA primer is added. So by using the 3' hydroxyl group of this RNA primer, what is going to be synthesized? Deoxyribonucleotides are synthesized in 5' to 3' direction by the help of the enzyme called DNA polymerase 3. Okay, so one of the template which is synthesized towards the fork continuously, okay, to form a leading strand. So one of the new strand will form continuous, in a continuous stretch is called leading strand, and the other template will get replicated discontinuously. The other template which is on the opposite side will get replicated discontinuously. Due to discontinuous replication here, that is going to result into a small many fragments, and those fragments are called Okazaki fragments. They are named behind its discoverer Okazaki. So the fragments which are formed due to discontinuous replication on the opposite strand or the opposite side template, they are called Okazaki fragments. Then such the discontinuously formed the strand is called lagging strand. The discontinuously formed strand is called lagging strand here. Okay, then this is going to be replicated away from the replication. Why? Now you may have the question here that why this strand is replicated discontinuously into small fragments? Because as I mentioned here, the replication will take place in only one direction, that is in 5' to 3' direction. Okay, it won't occur in opposite direction. So since this template was available in a continuous stretch, okay, so the continuous replication took place on this template. But this template, as it is itself in 5' to 3' direction, this is not available as in a continuous stretch. Hence, as it is stretched open into a small pieces or into a small patches, okay, so hence the complementary to that extent only the fragments of the DNA will get synthesized, and those DNA fragments are called here Okazaki fragments. Okay, now all these Okazaki fragments join together by sealing their sugar phosphate backbone by the enzyme DNA ligase to form the continuous stretch of a strand. Okay, now what happens after the, all the Okazaki fragments are joined here, the, what I said here, the another strand is formed as a leading strand. Both the two daughter DNA molecules will get formed from the single parental DNA. Two daughter DNA molecules will be get formed here. So entire process, once it gets over, now what happens, the next comes that DNA polymerase 1. So that is going to remove this. Since it is the entire DNA structure, there should not be any RNA primers here, so that once the entire replication is over here, the DNA polymerase 1 enzymes comes over here, and that is going to replace this all the primers. Here in the leading strand there will be only one primer is added at the beginning. Okay, but whereas if you take the lagging strand, each Okazaki fragments include a primer, so that all the primers from each of the Okazaki fragments are removed and replaced, and one from the leading strand, all together removed and replaced by deoxyribonucleotide triphosphates by the enzyme called the DNA polymerase 1. Okay, then what happens after all the entire process gets over? Once the DNA polymerase 2 enzymes goes around the DNA molecule and checks for wrong base pairs. Okay, if at all, means cross checking or proofreading enzymes, as I mentioned here already. So that is the proofreading enzymes. It will proofread and come, and if it identifies if at all there are any base or mismatching base pairs are found here, that is immediately going to be removed and replaced by the DNA polymerase 2 enzyme, okay, into the deoxyribonucleotide triphosphates. So this is how the entire replication takes place. Then finally, after each round of replication of one DNA fragment, you are going to found the formation of two daughter DNA molecules. So in each of the daughter DNA molecule, you can find one of the old strand or the template in the form of template, which, or you can say parental strand, which is conserved, and the other one is only the newly formed strand. As it is complementary to the template, we can also consider it as a complementary strand. So as in the newly formed daughter DNA, here one of the parental strand is conserved as it is considered as a semi-conservative method of DNA replication. So once again, please go through the entire mechanism of replication and understand properly, and be prepared for your competitive as well as board examination. Okay. Students, let us recall once again the entire mechanism of DNA replication. So starting from the definition, so it will be asked for 1 mark in the examination, define the semi-conservative method of DNA replication. As I mentioned, so semi-conservative method of DNA replication is a method of duplication of DNA in which the newly formed each daughter DNA molecule contain one parental strand and the other newly formed or newly synthesized strand. Since one of the strand of the parental DNA, that is out of 2 strands, means 50% you can say, out of the 2 strands will be conserved in the newly formed daughter DNA, it is considered as a semi-conservative method of DNA replication. Now the DNA replication, as I've mentioned, all the points we shall recall here, always begins at a specific point called origin of replication, and as soon as the origin of replication is detected by the helicases or amylases enzymes, they bind to it and they start moving along the double-stranded helix of DNA. As soon as they start moving along that, 2 strands of the DNA get separated to form a Y-shaped structure called replication fork. Then each separated strand, as I mentioned, it acts as a template on which the new or complementary strand is going to be synthesized. And the DNA replication, as I mentioned, it is nothing but DNA polymerization, and it occurs only in 5' to 3' direction by the major DNA polymerizer, the enzyme called DNA polymerase 3 enzyme. Then as soon as the DNA polymerase enzyme is going to detect the template, it won't begin the replication until and unless there is an addition of primer. Okay, so what is a primer? Primer is a short segment of RNA which is added at the 5' end of the template DNA to begin the DNA replication, and that primer is added or synthesized by the enzyme called primase or the RNA polymerase 1. So this is how the DNA replication begins. Okay, once the DNA replication begins, see what is the role of RNA or the primer here. I mentioned already, is the primer is going to provide the 3' hydroxyl group for the further DNA polymerization. So DNA polymerase 3 enzyme utilizes the 3' hydroxyl group of of the RNA strand here. With the help of that, it starts adding continuously the deoxyribonucleotide triphosphate, okay, in 5' to 3' direction. So this is how the DNA replication is going to begin. So once the DNA replication begins here, the one of the template which is in 3' to 5' direction, which is replicated continuously, so hence to form the leading strand, okay, and as the other template which is not available into continuous stretch, which is going to be available into a small fragments, so that the newly synthesized strand is also gets discontinuously replicated into a small fragment. So those fragments I mentioned already in the diagram that they are called Okazaki fragments, and such a new strand which is synthesized discontinuously, and that is called lagging strand here. Then those fragments are called Okazaki fragments, and all the Okazaki fragments join together with the help of enzyme called DNA ligase to form a continuous stretch of the enzyme. Then what happens after this? The RNA primers are going to be removed by the enzyme called DNA polymerase 1. So there will be only one primer in the one of the template where the leading strand is formed here, and there will be many more primers which are included in each of the Okazaki fragments. So all the primers are removed together and replaced by the enzyme called DNA polymerase 1. And finally the DNA polymerase 2 enzymes, after the entire process of replication gets over here, that comes over all around the replicated daughter DNA molecules and proofreads here, cross checks for the mismatching base pairs, and it repairs how? By removing the mismatching base pairs by introducing correct and matching type of base pairs, and that is done by DNA polymerase 2 enzymes. Hence it is considered as the minor repair enzyme, okay. So finally you can see the one more diagram which is just like what I have drawn on the board and explained already. So once again you go through these diagrams, and finally you can say here at the end of the replication, as the two daughter DNA from the single parental DNA molecule, two daughter DNA molecules will get formed here, and in each one of the daughter DNA molecule, one strand will be the parental strand and another strand will be the newly synthesized strand here. So since one of the parental strand is passed on to the newly formed daughter DNA, that would be equal to 50%. Hence semi means half. So this type of replication is considered as a semi-conservative method of DNA replication. Please go through. I hope you have understood. Twice I have explained here. I hope you have understood well. And please remember, each and every point of this is very, very important for the point of your competitive examination as well as for your board examination. Thank you.